short lab today. I think it's just the acid-base lab that you guys have. So in the lab, once you guys finish that, and I'm just gonna draw out sort of like a table listing the different type of acid-base imbalance for you guys to know. All you guys have to know is that particular table. Well, I mean, if you guys want to know more, read more into it, research more. But that's mostly the information that you need for the acid-base portion, right, of what the lecture questions I'm gonna ask you, or lab question I'm gonna ask you. Okay, so let's talk about the digestive system. Now, we have food that we eat, and the reason why we consume food is because we need to break that down. We need to deliver the carbon sources to our cells and tissues, and our cells and tissues will take in those carbon sources, they will break it down, and they will make energy from that carbon source. So we need to satisfy all of our cells and tissues, and by delivering those carbon sources, those cells are satisfied. The only way we can do that is to consume food ourselves, because we can't make carbon sources on our own, right? So therefore, the three main types of carbon sources that we eat as food is proteins, carbohydrates, and lipids. Okay, so that's it. So we'll talk about how those three are being processed. All right, so now for the digestive system, the overall structure of the digestive system is essentially, obviously, to digest the food, to absorb the food, and to do whatever you need to do to deliver that food to where they need to go. So here are some functions listed for the digestive system. One of its functions is what we call motility. Motility is movement. Okay, so a couple of terms for you guys to kind of keep in the back of your mind. The terms that you really need to know are the last two, but that term, the first term called ingestion, is essentially a term that refers to taking food into your mouth. Okay, and then once you put the food into your mouth, the next term is mastication, which basically means chewing. So therefore, once you guys, or if you've already gone through your anatomy, or you are going through anatomy right now, and they're talking about the muscles of mastication, that's the muscle you use to chew and break down your food. Okay, and then deglutition is essentially swallowing. Now, once you swallow the food into these particular tubes of your digestive tract, all of these structures are surrounded by, for the most part, smooth muscle. And smooth muscle is autonomic in terms of their activities, right? It's involuntary activities. So as soon as the food enters, let's say the esophagus, which is a large tube that conducts the food into the stomach, there's going to be a series of muscle contraction and muscle relaxation that allows for that food to move in one direction. So imagine you have a long tube. Okay, food enters that tube. The muscle group that is below that food is going to relax. The muscle group that is above that food is going to contract. So it's sort of like squeezing progressively to move that food downstream. Okay, so this differential relaxation and contraction allow for food to move, and as the food moves, the next set relaxes, and that set contracts. Next set relax, contracts, relax, contracts. So we call this peristalsis. Okay, it's a unidirectional movement of the food through the digestive tract, facilitated by differential contraction and relaxation of your smooth muscle group. Okay. Now, the other type of motility that is facilitated, that is being carried out within your digestive system, is what we call segmentation. Segmentation is akin to taking some liquid, put protein powder in there, and just shaking it up. Okay, so this is a mixing type of motility activities within your digestive system. So the muscles can contract and relax differentially to basically keep those food contents bouncing back and forth or mixed together within those organs, along with other secretions that's being produced by those organs, so that all the structures can mix together. Because in the secretion, let's say, of your stomach or of your oral cavity or anywhere else, it may contain digestive enzymes. And you need those enzymes to break down some of that food content into the simplest structure so you can absorb it, right? So these are just some movements. So here's peristalsis, basically contraction to push food down and contraction to push food down. So all the muscle down here is going to relax to allow that food to move progressively down this tube. Segmentation, let's say you have these pockets of food versus secretions, you want to mix them together, so therefore the muscle contract and relax differentially, kind of bounce the food back and forth in that area, and essentially allowing them to mix nicely together. Okay, so that's segmentation. All right. Second function of the digestive system is essentially for the purpose of secretion. In terms of secretion, there's two different types, right? We talked about this once upon a time. There's endocrine versus exocrine secretion. Exocrine secretion is secretion of products directly into a duct, and from that duct, those products will be secreted into the immediate environment. around that tissue or that organ. Okay, so let's say salivary secretion. When you secrete your saliva, there's a gland called the salivary glands that secrete the saliva into a duct called the salivary duct, and the saliva will be conducted to be secreted into your oral cavity. So that's the environment in which saliva is secreted. If you have digestive enzymes made by, let's say, the pancreas of your body, the pancreas will secrete that digestive enzymes into pancreatic ducts, and those enzymes will be delivered into the small intestine where they will work. Okay, so therefore, exocrine secretion is a secretion event that is facilitated by some kind of glandular tissue that secretes their product into a duct, a tube-like structure, and those products will be secreted out into the immediate environment of that tissue. Okay? Now, the other type of secretion we call endocrine secretion. In endocrine secretion, this grouping of glandular tissue, they don't have a duct associated with them because they've lost contact with the environment around them. So they're internalized into some kind of tissue, but they have a lot of blood vessel coursing through these cells and through this tissue. So therefore, when the endocrine glands secrete their products, they secrete it directly into the blood circulation. Okay? And then whatever those signals, those proteins, whatever those structures are that are secreted into the blood is going to be carried throughout the whole body, right? To work at distant site or some immediate site or whatever the case is. But endocrine secretion is secretion of hormones for the purpose of communication and things like that too. Okay? So keep in mind these type of secretion and where it happens along the digestive tract. Because as we talk about the digestive system later on, we're gonna all take a tour, right? We're all gonna get into a little school bus. We're gonna shrink ourselves down to a pill size. Somebody's gonna swallow us, and along the way, I'll be your tour guide. Say, oh yeah, over there, that's your oral cavity. Over there, that's your anus and stuff like that. Get ready to get shot out of that particular hole, right? All that good stuff, and then we rinse down the bus, and then basically, maximize ourselves back to our usual size. Okay. So along the way, I'm gonna show you where the secretion is happening. All right, the next function is called digestion, basically what the digestive system is for, right? Digestion refers to breaking down food particles. Now, there's two ways for us to break down food particles. One is a physical digestion, basically putting food into your mouth, chewing it, and breaking it down. That's a physical digestion. Okay? Or if the food enters, say, the stomach, and the stomach has this segmentation activities where it can continually churn and physically break down the food. That's also a physical digestion activity. Okay? The other type of digestion is called chemical digestion. In chemical digestion, this is facilitated by the enzymes that are secreted out by your glandular tissue. So, so if the glands are secreting the enzyme then will basically take that food particle and break the chemical bonds that hold that food particles together. And if you break that bond, we call that chemical digestion. Okay? So therefore, you need to be able to physically and chemically digest your food, because the next function is what we call absorption. And in absorption, the cells, mainly of the small intestine, is going to act to take in that simplest form of those food particles, which means all your carbohydrates must be digested or broken down into what we call simple sugar molecules or monosaccharides. All of your proteins must be broken down into amino acids. And all of your lipids, i.e. triglycerides, must be broken down into what we call fatty acids in order for the cells to absorb those contents into itself and deliver it to the blood or to the lymphatic circulation, okay? Because once you take in those contents of carbon sources, then those contents will be delivered throughout the whole body, or eventually throughout the whole body. Okay? So absorption is important. So that's the whole purpose of eating, right? So you can digest it, break it down, and you can absorb it into your body. All right. Fifth function of the digestive system is for the purpose of storage. So there's in certain areas of the body where you can store these particular carbon sources. In areas you can store it as a temporary means before you eliminate it altogether. And of course, the other function would be the elimination of undigested materials. Because a lot of the food that we take in, we cannot digest or we cannot break down completely because we don't have the enzymes that's there to break down those food contents. So let's say if you eat an apple You can basically process and digest some of the stuff inside the apple, but you cannot break down the skin of the apple, right? So therefore, you can mash it and break it down physically to a certain, texture, but then you cannot completely chemically break it down because we don't have the enzymes to break down the cell walls of that covering of the apple, or of the vegetations and stuff like that, right? So a lot of times those things will act as fiber material to help us, easily get rid of the undigested materials, right? Make our fecal materials a little bit softer and stuff like that, and not super hard, right? All right, so that's storage and elimination. Now, along the way, throughout the digestive tract, there are certain areas within the mucosa surface or close to the top surface of these particular structure that has high collections of white blood cells. And these particular area, because it has a lot of white blood cells, form some kind of immune barrier, okay? So therefore, the immune barrier in the digestive tract, we refer to as the Peyer's patch, named after Dr. Peyer. But collectively, any area within any internal organ that has a high collection of white blood cells are known as the mucosa-associated lymphoid tissue. Okay? It just so happens that the mucosa-associated lymphoid tissue in the digestive tract is referred to as the Peyer's patch. Okay? All right, here's all the anatomical structures that make up your digestive system. So think about the digestive system mainly consisting of all these particular organs that are attached together. And when they are attached together, they basically just form a long tube that is continuous from one end to the other. Okay, so we're just a convoluted earthworm, so to speak, right? If you stretch out our mouth all the way to our anus, basically it's just a long tube that is continuous, right? So all these tubings, we collectively call the gastrointestinal tract, okay? The gastrointestinal tract can be referred to as the GI tract or the alimentary canal. It's roughly about 30 feet long in length. It starts with the mouth, which is called the oral cavity. And from the oral cavity, you divert that food into what we call the pharynx. And the pharynx, there's two portions. There's an oral pharynx and a laryngopharynx. From there, food is diverted into a long muscular tube called the esophagus. From the esophagus, the food is going to be conducted all the way down into the stomach. The stomach is located in your abdominal cavity in what we call the left abdominal quadrant. So you take your abdominal cavity, you cut it in 4 quadrants, where you draw a line down, you draw a line right at the umbilicus. It will separate into a right upper, right lower, and left lower quadrant of your abdominal cavity. Okay? So the stomach is located in the left upper quadrant. And from there, the stomach empties the food into the small intestine, which has 3 parts. There's the duodenum, the jejunum, and the ileum. Okay? Essentially, the small intestine is the longest organ of your GI tract, of your gastrointestinal tract. From that, the ileum will deliver undigested food into the large intestine, which includes the cecum, ascending colon, transverse colon, descending colon. And as the colon descends on the left side, it's going to form an S-shaped structure called the sigmoid colon. Sigmoid colon then straightens out into a pouch we call the rectum. The rectum then narrows down into a small canal we call the anal canal, and the anal canal terminates as the anus, which then opens to allow for fecal material to be evacuated out of the body. Okay? So all of those structures makes up what we call the gastrointestinal tract. Along the way, we also have accessory organs that will aid in the process of digestion, okay? So in the oral cavity, you have your teeth, your tongue, and your salivary glands to kind of help break down some of that food, both physically and chemically. In the abdominal cavity, once you get the food in there, there's an organ called the liver, and the liver is one of the largest singular organs that sits in the right upper quadrant of your abdominal cavity. Okay? The liver is amazing because it has a lot of different functions for your body. So the liver itself is going to process all that food, and there's a structure underneath the liver called the gallbladder, which essentially is going to store the substance we call bile. We'll talk about that in a second. And of course, inside the abdominal cavity, there's this organ called the pancreas. The pancreas basically will produce every single digestive enzymes, pretty much every single one, that is needed for the chemical digestion of the food in the small intestine. Okay? Furthermore, the pancreas also is part of the endocrine system as well, because it has these collection of cells that constantly secrete hormone to regulate your blood glucose. Okay, we'll talk about that when we get to the endocrine system. All right, so here's all the anatomical structure. So here's the oral cavity. Okay, here's the oral pharynx back here. Here's the laryngeal pharynx down the esophagus, into the stomach, into the small intestine. So the first portion of the small intestine is called the duodenum, or the duodenum, doesn't matter how you say it, just spell it correctly, right? Then it goes into the jejunum, and then all the way into the ileum, which then dump that undigested food into the cecum, and then that food is going to be transported up the ascending colon because it's moving upward, and it makes a sharp turn here to go from the right side to the left side, so we call that the transverse colon. And then from here, it makes another 90-degree turns to go down, so we call that the descending colon. And it makes this S-shaped turn called the sigmoid colon. Once down here, it enlarges into a pouch called the rectum. The rectum is where we temporarily store waste material of the digestive tract. And then from the rectum, it's going to narrow into the anal canal and then exit through the anus itself. Okay, so those are all your alimentary canals. Keep in mind they're continuous on both ends. And the contents inside of your digestive tract is considered to be the external environment itself. Okay? The only time you consider something to be internal or is part of you is if it's inside your cells or inside your blood. Otherwise, it's still part of the overall external environment. Okay? All right, now if you take the digestive tract and you dissect it out, you will see distinctly 4 layers that make up your digestive tract. The innermost layer is called the mucosa. The mucosa contains some kind of secretory or absorptive epithelial tissue that allows for you to secrete things or absorb food, deeper into your body and things like that. Okay? The mucosa basically sits on some kind of basement membrane, which is a connective tissue, and surrounded by a very thin, smooth muscle layer. Right? The next layer down, or the next layer below the mucosa, we call the submucosa. In the submucosa, it contains a huge amount of connective tissue, so it's a thicker layer of connective tissue. It contains larger nerve vessels, large lymphatic vessels, and larger blood vessels. This is where you deliver all of these digested food from your intestine into the submucosa. So once you deliver it into the submucosa, that food then enters either your blood or your lymphatic circulation, and that will take all that content to where you need to store them. Okay. Now the third layer, the layer that's outside of the submucosa, is a thick, smooth muscle layer, typically. Right? We call this the muscularis externa. And because it has smooth muscle, typically it has 2 layers of smooth muscle that overlaps each other. And because they overlap each other, they will contract in different orientation. And by contracting in different orientation, it can either move the food downstream or allow for mixing of the contents within those particular structures. Okay? And finally, all of this is then covered by a connective tissue layer called the serosa, which is just basically an outer connective tissue layer that protects all of these vital organs. All right, so if you look at it, this is what it looks like. So here's the innermost layer called the mucosa. The mucosa is the lumen of your digestive tract, so the food is traveling within that lumen. You can see that there's a bunch of epithelial cells here, right, that interacts with the contents of the food that will act to absorb that food eventually, or to secrete certain things out to kind of mix with that food content. All the epithelial cells are bound to a thin basement membrane and surrounded by a very thin layer of smooth muscle. So all of that makes up your mucosa. Okay. Next layer, the layer that's below the mucosa, we call the submucosa. And sub means underneath, mucosa is the mucosa. And this submucosa contains a thicker connective tissue layer that also contains nerve fibers and blood vessels, so arteries and veins, and also lymphatic vessels as well. Okay, that's part of your lymphatic circulation. Okay, so the food that is being absorbed from this area will be diverted and delivered to those blood vessels and lymphatic vessels. Okay, so they can transport that to wherever you need to store that food content. Okay? The third layer we call the muscularis externa, and this is a thick, smooth muscle layer. You can see there's two distinct smooth muscle. One is oriented circularly. So imagine this is the length of your digestive tract. The innermost layer, the inner layer of that smooth muscle is circular, so it's perpendicular to the length of the digestive tract. Okay? Whereas the outer smooth muscle layer is longitudinal, so it's parallel to the length of that digestive tract. So therefore, the inner layer is circular, it contracts this way. The outer layer is longitudinal, it contracts this way. Okay, so therefore, there's two different sort of like orientation of contraction that's happening. And then finally, the outermost connective tissue that kind of protects all of these structures internally is your serosa. Okay. Now keep in mind, you also see a bunch of nerve complexes here. Okay, we'll talk about these nerve complexes in a second. All right, so there are ways to kind of stimulate the activities of the digestive tract. There's an extrinsic way, which means there's an external way, and the main way to externally or extrinsically stimulate the digestive tract is through the autonomic nervous system, i.e. the parasympathetic division, right? Because remember, if you're trying to rest and digest, you need the parasympathetic nerve and not the sympathetic. Because when you try and run away from danger, the sympathetic nerve kicks in and inhibits the activities of your digestive tract. But when you have time to kind of relax, cook your food, digest or eat the food and digest your food, then it's a parasympathetic that is the main autonomic division that regulates that. So therefore, in terms of the digestion itself, it's the vagus nerve, which is your cranial nerve number 10, right? And also the pelvic splanchnic nerve, which comes from the sacral region of the spinal cord. Sympathetic. Now, in addition to that, you also have a bunch of hormones that can be secreted out too, because there are some cells and glands that is regulated by this endocrine activities imposed by the hormones secreted out by endocrine glands. Okay, and we'll talk about those structures, later once we get to it. Now, along your digestive tract, there's also a bunch of what we call intrinsic cells. Intrinsic cells are built in to that particular organ itself. So like in the heart, the intrinsic cells will be your SA node, your AV node, and your Purkinje fibers, which contains autorhythmic cells, right? So there are also autorhythmic cells in the digestive tract. And these autorhythmic cells are located mainly in areas that has a lot of smooth muscle that they need to control to move the food, to mix the food, and things like that. So these particular interstitial cells, intrinsic cells, we call the interstitial cells of Cajal, okay, named after Dr. Cajal. So basically, these cells have the ability to also produce action potentials to regulate the secretion that's going on inside your digestive tract and also to regulate the smooth muscle contraction and relaxation. So, in addition to the interstitial cells of Cajal, you also have this overall nervous system that is dedicated to the digestive process itself, and we call this the enteric nervous system. The enteric nervous system is actually made up of two networks of neural tissue. One is called the myenteric plexus, and the other one is called the submucosal plexus. So let me backtrack a little bit and go back to this picture, right, that we showed before. So the submucosa plexus is located within the submucosa. Hence, we call it the submucosa plexus, right? But the submucosa plexus also has a different name because it's still named after the person who discovered it, and it's also referred to as Meissner's plexus or the plexus of Meissner, named after Dr. Meissner. Now, within the muscularis externa, between the inner circular smooth muscle layer and the outer longitudinal smooth muscle layer, is another neural network. So another network of nerve tissue. And we call this one the myenteric plexus. So anytime you see the word MY or MYO, that refers to muscle. So this particular neural group is located within the muscular layer of your digestive tract. Okay? Between those two muscle layers. So the myenteric plexus is also referred to as the plexus of Auerbach, or Auerbach's plexus, okay, named after Dr. Auerbach, the guy who found that.
particular plexi of nerve tissue. Good? Any questions about the anatomical makeup of your digestive tract? So 4 distinct layers, right? You guys should be able to see those 4 distinct layers. Well, I mean, it's not histology, so you may not really care about it. Right? I'm gonna show pictures about it. Okay? But if I ask you to organize these things, like where the smooth muscles are located, where the myenteric plexus is located, where the serosa is located, you should be able to organize it in a way so that you can place all of these tissue together to form your digestive tract. Okay? All right. This is the action potential produced by the interstitial cells of Cajal. I think we're pretty much action potentialed out, right? So we don't need to know this. All right. And if you guys want to know, the main type of neurotransmitter that is stimulating most of your digestive process is acetylcholine, because parasympathetic activity utilizes mainly acetylcholine. Between the pre and the post ganglionic neurons, and between the post ganglionic neurons and whatever those effectors tissues are. All right, let's take a trip. So the rest of it I'm going to draw for you guys. This way, that's all you need to know now, everything I've drawn on the board for you. If you want to, you can obviously look back at some of the lecture stuff just to kind of like fill in the gaps, fill in the blanks. If there are any blanks what you guys are studying. All right, so let's take a look. So, food that we consume, right? We have carbohydrates, we have protein, and we have lipid. Proteins. I think carbohydrate, I don't know, is there an S? Carbohydrates. Different form of sugar, I guess. Okay, fine. Carbohydrates, proteins, and lipids. Okay, we consume this and it enters the oral cavity. So here's the oral cavity. Now the oral cavity is lined by a type of epithelial cell we call non-keratinized stratified squamous epithelium. Now, why do you guys think we need this thick layer of cells in the oral cavity?
Protect from abrasion.
Protect from abrasion. Why is there abrasion? What are we afraid of?
The food is rough.
Of course. You ever eat stale bread?
No.
Or any bread for that matter, sometimes it's pretty hard, right? Thanksgiving's coming up. Chew on those croutons for a little bit without kind of like letting your saliva, kind of break it down a little bit. Those things are hard. If you take in a protein bar, right? Let's say if you get one of those protein bar tonight, if there's some available, like and you guys are chewing on it, it's rough. If you eat a carrot, celery, any kind of vegetation, it's rough. So therefore, there could be some degree of sort of like friction or irritation that damage some of that mucosal surface. So because of that, you need that protection, okay? But it should not be your skin, so therefore it's non-keratinized instead of keratinized. Okay. So now, you take the food into your oral cavity. Oh, did you guys know there's I forgot what it's called. Maybe it's like the Mastication Society or something like that, or the Digestive Society of America, I don't know, whatever it is. They do recommend you chew your food a certain amount of time, okay? How many times, do you guys ever count how many times you chew your food before you swallow it? You're supposed to chew your food, I believe, at least 20 to 30 sometimes before you swallow that food, okay? Any kind of food, well, maybe except for like really soft baby food and stuff like that. But otherwise, yeah, you should be able to, you should chew your food that recommended amount of time to adequately physically break down that food content. So when you swallow it, you actually can break it down chemically better into the simplest form. Okay? And I'll tell you why that is in a second, and that's why it's recommended. Anyhow, okay, so the food enters the oral cavity. Okay. Once the food enters the oral cavity, your teeth and your tongue help to physically break this down, right? As you chew and tight and bare and grind your food with your teeth. Your tongue smashes that food against the roof of your mouth, against your cheeks and stuff like that. So you're physically breaking down that food. But inside your oral cavity, or actually outside of your oral cavity, you have a bunch of glandular tissue. And these are called your salivary glands. And the salivary glands will secrete saliva to mix with the food. And once the saliva mixes with the food, it forms a content called bolus. Right? So go home, if you want to see what your bolus looks like, go put food in your mouth, chew, and then stick your tongue out, look in the mirror, that's bolus. Right? It's just like a geo type of, food, broken up food particles mixed with saliva and all that good stuff. Okay? So the saliva helps to kind of break down that food into that bolus. Do you guys remember what the salivary glands are that we have? Okay, so sublingual, underneath your tongue. What else? Submandibular, underneath your mandible. Did you say carotid?
Carotid.
I don't know how to pronounce it.
Parotid gland. Good. So sublingual, underneath your tongue, you reflect or stick your tongue up, showing the ventral surface of your tongue. You can see that there's some areas sort of like larger in the midline of your tongue. That's the sublingual glands. The submandibular glands, if you trace your mandible, here's the angle of the mandible, down slightly below the angle of the mandible, and you press inward, you can actually palpate your submandibular gland. Okay? And the parotid glands are located right in front of your ear, right on top of the ramus of the mandible or the arms of the mandible. So basically right around this particular area. Okay. They will all secrete saliva into the oral cavity, right? And then that saliva will mix with the food to become bolus. Now some of these glands will also secrete digestive enzymes, okay? So say also secrete what we call digestive enzymes. So saliva, and they have this. They have a digestive enzyme. Now this enzyme, I'm just gonna write down the name of this enzyme, is called salivary amylase. And amylase is an enzyme specifically there to break down carbohydrates, break down complex sugar molecules. Okay? So all my enzymes, I will box it in blue. All my hormones, I'll circle in red. So therefore, the salivary glands that secrete saliva and this enzyme called salivary amylase, these are all exocrine glands because they secrete these substances directly into the oral cavity itself. Okay? All right. So what's the function of salivary amylase? Let's put it down. Okay, so remember, it's chemical, so chemically digest complex carbohydrate into what? Less complex carbohydrate, because we don't chew enough. And there's no way we can really break down all of these complex carbohydrate into simple monosaccharides. Okay? So less complex carbohydrates. And of course, sometimes, we have less complex carbohydrate that we consume, so we may potentially be able to break it down to that. Right? So less complex carbohydrate. And also keep in mind, the bacteria that lives in our oral cavity loves these less complex carbohydrates too. So if we, break down these things and we don't wash it off, adequately, and we allow that carbohydrate to kind of persist inside the oral cavity, those bacteria will start to break down those carbohydrates into sugar molecule, into simple sugar molecule, which they love to ferment, causes a high degree of acidity and therefore wear down on the enamel of your teeth, causing, you're just financing the dentist to buy his new car. Okay, so anyhow, all this is happening. Now, what do you guys think are the regulation of these particular glandular tissue What nerve regulates the secretion of saliva and salivary amylase from these salivary glands? Facial does what? We talked about this, right? In the autonomic nervous system lecture. Hypoglossal moves your tongue. That's what hypoglossal does. It's a motor nerve that moves the skeletal muscle of the tongue, not tongues. What do you think? Is this part of sympathetic or parasympathetic? It's rest and digest, right? So parasympathetic. And what are the 4 cranial nerves that participate in parasympathetic? Oculomotor nerve, that's number 3. Facial nerve, 7.
7, 9.
Glossopharyngeal nerve, number 9. And vagus nerve,
10.
number 10. Okay? So for the sublingual and submandibular gland, both of those are controlled by your facial nerve, or cranial nerve number 7. Parotid gland is controlled by your glossopharyngeal nerve, which is cranial nerve number 9. Okay? Alright. If you guys don't know that, it's okay. It's a couple points. Not that big of a deal. Thirsty. Okay. Now this bolus, once it has been adequately chewed and physically broken down, and some of the salivary amylase has now broken down some of the complex carbohydrate into less complex carbohydrate, the bolus now enters your pharynx and eventually enters your esophagus. So let's go to the esophagus. So the esophagus, as bolus enters, the esophagus is this long tube, right? So this bolus enters, and with the esophagus, there's going to be a series of muscle contraction and muscle relaxation, so that you can move this bolus down. So we call that segmentation. Sorry, peristalsis. Okay. Now, the important thing, or the very unique thing about the esophagus is that, first of all, the mucosal surface of the esophagus is still lined by the same type of epithelial tissue. So we'll say non-keratinized, stratified squamous epithelium, right? Because you still need that protection from all of that food contents that you are now delivering into the esophagus itself, okay? So, non-keratinized stratified squamous epithelium, food moving down. Now that's the mucosa surface, so all through the esophagus is lined by the same type of epithelial tissue, it doesn't change. What changes in this esophagus is that the muscle, the type of muscle in the muscularis externa, that muscular layer of the esophagus, is quite different. So in the upper third, for the most part, so let's say if you divide the esophagus into 3 regions, in the upper third, of your muscularis externa, that is lined by skeletal muscle. Okay. The middle third, this way, middle third, lined by a combination of both skeletal muscle and smooth muscle. So more smooth muscle than skeletal muscle, okay? And the lower third of your esophagus is lined exclusively by smooth muscle. Okay. So what does that mean? It means that when this bolus enters the top of your esophagus, you can actually voluntarily contract that skeletal muscle to regurgitate that food back out. I don't highly recommend it because it does feel very uncomfortable to do so, right? It's not like throwing up, it's more like you're inducing that. I can't even do it. Something to that effect, right? It's sort of like you take some tonic in, you take some food in, you're like, this doesn't taste good. I don't know what it is. I think like, you know, my wife's trying to poison me, or whatever the case is, or something's happening, this is spoiled, whatever the case is, so I'm gonna throw it back out, or I'm gonna eject it back out, right? So you can do that. But once the bolus reaches the middle third, then it's harder for you to do so, because now there's more smooth muscle there. So it becomes involuntary, and that food will reach the stomach eventually. Okay. Now, of course, you have skeletal muscle on the outside, so you can actually increase that contraction of skeletal muscle on the outside to force that food out, but still, not something that would be recommended in the long run. All right, good, so that's the esophagus. All right, so here comes the bolus. So now as the bolus leaves the esophagus, it goes into the abdominal cavity. So you guys remember the diaphragm, which is that sheet of skeletal muscle that basically physically separate the thoracic cavity on top and abdominal cavity on the bottom. There's 3 holes. One of those holes is for the esophagus to pierce through, okay? Pierces through into the abdominal cavity and connects with the stomach itself, all right? So let's see. Guess we do it here. Sorry, we're gonna have to split this. So here's your stomach. Now, once you move into the stomach, the mucosal lining of the stomach changes in terms of its epithelial tissue. So instead of non-keratinized stratified squamous epithelium, the stomach is lined by simple columnar. Let me draw out the sort of like the structure of the stomach. So imagine, and the stomach has all these convolutions and stuff like that, and all of the cells of the stomach are simple columnar epithelium, and they have these deep little fold, and we call this the gastric pit. Okay, so in here, this is what we call the gastric pit. And the environment of the stomach itself is highly acidic. It is one of the most acidic area within the body. It has a pH of about 2.5. Okay, so very, very acidic, which I don't recommend sticking your finger in there, even if you can. It's detrimental to the tissue of your finger. Okay, so draw a bunch of these particular cells. These are all columnar cells. I'll accentuate some of these cells because they are important for our discussion. So down here, so at the bottom of the pit, you have this cell that's slightly bigger, okay? And kind of right next to it, you have another type of cell here. And slightly above that, you have another one that is slightly more bulbous, and then up here, still, these are still deep within the gastric pit, right? It's not like close to the top. But as you get closer and closer to the top, right on the top here, you have these cells out here, right at the top of that gastric pit. Okay? All right, so that's the makeup of this. Now, some of these cells are endocrine cells, and some of these cells are exocrine cells. So therefore, keep in mind which one they are. Okay, the cell right at the bottom of the gastric pit, we call this the G cells. Okay. So here's the gastric pit. Let's say the bolus enters. Bless you. Bolus enters into this gastric pit from the esophagus. Now, once the bolus enters, there's going to be a stretching of the stomach to accommodate that entrance of the bolus. So the stretch is going to stimulate the G cells. So stretching of the G cells due to the incoming bolus is gonna stimulate, sorry, stretch of the stomach due to the incoming bolus is gonna stimulate that G cell, okay? Now I'll just draw it. It's okay, I'll erase it later. Make sense? Okay, so that's one of the way to stimulate the G cells. The second way is, let's say if you stimulate it using the vagus nerve. So the vagus nerve has a direct stimulation to the G cells as well. Okay. A third method to stimulate the G cells is basically a way to prepare yourself for eventual incoming food bolus, and we call that the cephalic phase of eating. Okay. The cephalic phase essentially is a phase where you're imagine you guys are hungry, like right now, you're hungry because you just came from work or whatever the case is, right? And you haven't really had time to have dinner and stuff like that. So you're sitting there, and all of a sudden Anton like breaks out a nice juicy piece of steak. I don't know where the hell he got that steak from. You need to question the man because I mean the cafeteria is already closed. You shouldn't be carrying steaks around like that. It's dangerous stuff, right? He opens up that piece of steak, and it's like the smell starts to percolate throughout the room. You know, like Tamara's sitting over there, she's like smelling. I was like, damn, this is pretty good. That makes me hungry, right? So when you smell the food, that is the cephalic phase of eating. So when you're hungry and somebody talks about food, you're like, oh man, I'm getting really hungry just listening to this damn conversation. That's the cephalic phase of eating. If somebody put pictures of food on the wall or on the screen, and you're like, wow, that looks really good, right? Like restaurants, when they take pictures of all the dishes and stuff like that, that's to incite that type of reaction. Okay, so that's all what we call the psychological phase to prepare yourself for eventual digestion of the food itself. Okay, so therefore, you think about it, all of these particular activities here is going to stimulate the G cells to do what it needs to do, right? So therefore, during that time, your stomach starts to gurgle a lot. There's a high activities within the abdominal cavity. So the G cells, in response to any of these stimulation, it's going to secrete a hormone. So therefore, the G cell is an endocrine cell, okay? It secretes a hormone. And this hormone is called gastrin. Okay. And what gastrin does is that it gets into the blood. It doesn't have to travel that far, because all it does is gets into the blood and then stimulate this next cell over here. And this neighboring cell is called the entero. Entero means digestive system. Chromaffin. Chromaffin means a colorful cell. But this is a chromaffin-like, because it looks like a chromaffin cells. It acts almost like a chromaffin cells, but it's not truly a chromaffin cell. But this chromaffin-like cell is located in the digestive tract, hence enterochromaffin-like. And these guys are cells, so enterochromaffin-like cells. Okay. And as gastrin stimulate those enterochromaffin-like cells, these cells are also endocrine cells. So they will also secrete their hormones. And the hormones secreted by the enterochromaffin cells are histamine. Okay. And the histamine is going to stimulate this next cell. And this cell right here is now referred to as your parietal cells. Okay. The gastrin that is secreted by the G cells can also act on the parietal cells, but it does so as a minor stimulus to the parietal cells. The major way to activate and to regulate and to stimulate the activities of the parietal cell is through histamine that is secreted by your enterochromaffin-like cells. So we'll draw a little bit of gastrin here as well. Okay, so this is the major method. This is the minor method, or minor stimulus, major stimulus to the parietal cells. Okay? Now, once the parietal cell receives this particular stimulation from histamine, because there's histamine receptors on the parietal cell for the histamine to bind to and act on, parietal cell is an exocrine cell. So it will secrete hydrogen ion and chloride ions. And the hydrogen ions and chloride ions that's secreted into this environment of the stomach will combine together. So it combines together to form hydrochloric acid. Okay? And when formed hydrochloric acid, it's going to decrease the pH and maintain it at about 2.5. Make sense? Okay? So this is the acid that basically allows for this environment to be highly acidic all the time. Okay? That's one of the things that the parietal cell does. It's an exocrine cell because it secretes these directly into this environment. It doesn't secrete this into the blood, it secretes it into the lumen of the stomach itself. So therefore, the gastric pit and all the lumen of the stomach is highly acidic at this point, right? Has a pH of about 2.5. Okay. Now up here, you have another set of cells, we call these the chief cells. And the chief cells can be stimulated by gastrin. Or more importantly, it is actually stimulated by the vagus nerve. And the stimulus to the chief cell by the vagus nerve and the gastrin is equal, relatively. Okay? So the strength of the stimulus to the chief cells coming from two different sources, pretty much the same. Okay? Now when the chief cells are stimulated, it will secrete an inactive enzyme. Because the chief cell is also an exocrine cell. And this inactive enzyme is referred to as pepsinogen. Okay. The pepsinogen is converted into its active form if and only if you have adequate amount of hydrochloric acid. Okay, so the acidity inside the stomach actually activates this pepsin. And when this pepsin gets activated, it becomes, this pepsinogen gets activated, it becomes pepsin. Okay? So pepsin is a digestive enzyme. And the job of pepsin is to chemically digest complex proteins into less complex proteins. Okay, so the function to chemically digest complex proteins into less complex proteins. Does that make sense? There's not enough time in the stomach for it to digest this complex protein all the way down to its simple amino acids. Okay?
Does hydrogen chloride also have a breaking down effect?
Not necessarily. Because with that acid, it may have some degree of, let's say, denaturation and stuff like that, but not drastically in that way, right? You do need this enzyme to basically chemically break that down because yes, we are trying to break down this protein, and that denaturation kind of helps along the way, right? But pepsin is doing most of that job. Okay. Good? Yeah, so far so good, right? Okay, so now you have to ask yourself, and there's other things that's being secreted too that I didn't really talk about in this particular scenario. I just want you guys to kind of focus on this main stuff that's going on. Okay? Now, if the stomach itself is highly acidic, how is it that these cells don't get destroyed? Because these cells are not like super cells, they're just normal columnar cells, right? If the pH is super acidic, the environment is super acidic, then those cells should basically die as well. Luckily for us, we have these cells up in the top of the gastric pit, and we call this mucous neck cells. And the mucous neck cells are stimulated by the vagus nerve as well. And when the vagus nerve stimulate these mucous neck cells, they will secrete a thin layer of fluid and mucus that covers the surface of all of these cells. Okay, so layer of mucus. And this layer of mucus contains a lot of bicarbonate ions. Okay, so layer of mucus with bicarbonate. And bicarbonate is an ion that acts as the main buffer for the amount of acidity and alkalinity that we may have, okay? So therefore, the overall environment of the stomach itself, including the gastric pit and the rest of the stomach, is highly acidic. But right at this interface here, as the acids tries to penetrate and destroy these cells, the acid is neutralized by that bicarbonate. And therefore, the environment immediately on top of those cells are neutral. And therefore, these cells are left unharmed. Okay? Now, we constantly have a battle within our body between how much of these bicarbonate in this mucus is secreted versus the amount of acidity that exists within the stomach. If you are basically taking some kind of external medication or external, I don't know, whatever it is that you consume that decreases the amount of bicarbonate or increases the degree of acidity, because they're balancing each other, right? So therefore, if you start to, for some reason, decrease the bicarb, then the acid will start to wear away this mucus layer and start to destroy these cells. If you have high bicarb, but now all of a sudden you introduce something that has a higher degree of acidity into your stomach, then that higher acidity will overwhelm your bicarb system and start to wear down this particular lining, okay? Now, of course, if you do it one time, it's not a big deal, but if you constantly subject yourself and your body to high acidity, let's say if you drink soda every single day, like twice a day or whatever it is, every single day for the next 2 months, I assure you, you will start to wear down this bicarb layer, okay? If you take a lot of aspirin or NSAIDs like ibuprofen and Advil and things like that for things like headache or whatever the case is, right? Then it will start to wear down this bicarb, because some of the contents of those medication will inhibit the production of bicarbonate. And therefore, now the acid is gonna start to win because there's a lot more acid that retains and less bicarb that's being produced. So because of that, then this bicarb layer starts to be overwhelmed by the acidity. If you are stressed, for whatever reason you're stressed for, maybe, I don't know, minorly is probably this class, and you know, you have other things to worry about, right? This class is the least of your worries at this point. But let's say you're stressed out because of something. You got 2 jobs to manage, you have 3 kids and 2 husbands and 2 whatever the case is, you know, 8 dogs you gotta walk every single day, and then you have to like pay the bills on time and do all this stuff on time. You're getting harassed by like, you know, the IRS because you haven't paid your taxes or whatnot. Like your teachers, you have teachers like me that constantly demand that you spell correctly and all that good stuff. Otherwise, I take your points away. So you're very stressed out psychologically and physically. So when you're stressed out, you actually make less bicarb. And because of that, now the acidity has a better chance of wearing down these cells and causes these cells to die. When these cells die and basically the acid wins and start to kill these cells, we call that an ulcer. Okay? And the reason why ulcer is painful is because as you erode away this cell, you also start to affect all the tissue in the submucosa area, where you have nervous tissue, all these nerves. So your pain receptors located in your submucosa becomes stimulated. And because of that, now you feel a huge amount of pain. The worst thing is that as you start to wear down these cellular layers into the submucosa, you also start to break blood vessels. Once you break the blood vessels, now you have active bleeding into the stomach itself, okay? So therefore, manage your stress, otherwise you're gonna have ulcers. You think it's stressful now, wait until you get into grad school. It's gonna be 10 times as stressful, right? You still have the same issues you had before, family obligations and all that stuff. Now, on top of that, 4 classes, 5 classes, that's this caliber. And keep in mind, if you fail, they kick you out, right? Maybe they'll give you 1 chance, 2 chance, or whatever it is, but they don't care. They already got your money, okay? So anyway, going back to this happy little thing called the stomach. All of these secretions that's being secreted out mixes with the bolus. So all of these collectively we call the gastric secretions, right? All the gastric secretions mixes with the bolus to form this compound called chyme. Okay, and chyme is highly, highly acidic, obviously, because it has a lot of acidic contents produced by that gastric secretion. Okay, so far so good. Any questions about our journey? All right. Keep your hands inside, don't stick it out. You're gonna get, eroded away by that acidity inside the stomach. Yes, Liz?
If you like take a bunch of like antacids,
You can.
like what would that be like?
Well, the antacid has the bicarb. So the bicarb helps to neutralize some of that acidity. Right? Now, if you take too much, then what happens to the acidity inside your body? It's too low. Now, well, it decreases the acidity, which basically now the pH starts to climb. So now you get into a process or a condition we'll talk about later called metabolic alkalosis. Right? So instead of the pH being maintained at whatever it should be, now the pH is slightly higher than what it should be. And of course, we're talking about consuming a huge amount of antacids for a long period of time. So you're popping antacids like it's freaking candy on Halloween and stuff like that, right? And you do it for like 2 weeks continuously. Then yes, you will neutralize a lot of that acidity. Okay, so yeah. Anything you put in the body, make sure you understand that you cannot do it consistently for a long period of time. The only thing that you actually put in the body all the time is food and water. That should be the only thing. Right? And within the food and water, you have adequate amount of vitamins and minerals already that satiate all the cellular process of the body. Okay, good. So, stomach. Now from the stomach, that chyme will leave the stomach to move into the small intestine. So, small intestine. Now keep in mind, the small intestine is where you will chemically digest all of your food. Or at least ideally, that should be the place where you chemically digest all your food. If you cannot digest completely all of those contents of the food that you have already physically and chemically broken down inside the oral cavity, and physically and chemically broken down inside the stomach, if you cannot break all of that stuff down to its simplest form in the small intestine, the cells of the small intestine cannot absorb that. And if the cells of the small intestine cannot absorb that, the cells of the large intestines will not absorb any of that stuff. And therefore, you will lose all of that food as feces. So you eat $100 steak. Let's say you decide, you know what, I'm gonna celebrate because I already finished whatever I need to finish. I'm gonna take my ass to Fleming's or Alexander's Steakhouse or whatever. I ordered like their economy grade steak, which is $150 or whatever the case now, okay, with all the fixings and the trimmings, and you're chewing that steak and you're eating and you're like, oh, this is so good, and you don't chew it adequately and you swallow it. Well, guess what? You're gonna have about $100 worth of poop and $50 worth of protein that you can actually absorb. Okay, so therefore, if you don't chew your food adequately, you will waste all of that food as fecal material.
So that's just for the protein and carbohydrates, right?
So far, we've only talked about protein and carbohydrate digestion.
Okay. Yeah, but what you're saying like right now about it like going to waste is only for the protein and carbohydrates.
Yeah, because you have to physically break down those proteins,
Yeah.
right? Let's say in the mouth. Yeah, so that when it gets to the stomach, then all of these acidity can also help further break that down, and all of these pepsins will chemically break down some of that. Just to prepare it, right, into smaller complex proteins so that the small intestine can eventually break it all down into amino acids and stuff like that. All right, so in the small intestine, it is kind of continuous with the stomach, so therefore the lining of it is still simple columnar. So simple columnar epithelial tissue. Okay. imagine now your chyme enters. Chyme's highly acidic, so therefore there are these cells inside the small intestine we call the duodenal cells. So these are columnar cells. Okay, because the duodenum or the duodenum is the first portion of the small intestine, the portion that is connected to the stomach, and is also the most important portion of the small intestine. It just so also happens to be the shortest segment of the small intestine. So these are called duodenal cells. Okay. The duodenal cells detect the acidity of the chyme. And some of these duodenal cells that's located right next to the stomach are also endocrinic cells, which means they have the ability to produce hormones and secrete that hormones into the circulation. So they will secrete a hormone. So collectively, I'll just pick them, this cell right here. They'll secrete a hormone called secretin. And there's a lot more hormones going on in here too, right? I just want to like summarize these ones for you. Secretin will stimulate the portion inside the pancreas, so it goes to the pancreas. And the pancreas has all of these cells in there that's part of this digestive process. And these cells produces and secrete all sorts of enzymes for you. And those cells are called the pancreatic ductal cells. But the pancreatic ductal cells are connected, the pancreatic acinar cells. And the pancreatic acinar cells are all attached to the ductal cells. So in the pancreas, it has this, the orientation of it is like this. So you have a bunch of these secretory cells, right? And they're all connected to a duct. Okay, and this duct is out here like that. Okay, and all of these cells over here basically is going to secrete things into this area right here. Okay. So these cells are called the pancreatic acinar cells. So these are acinar cells of the pancreas. These cells are called the pancreatic ductal cells because they form the ducts. Okay. So secretin will stimulate the pancreatic ductal cells. And when it stimulates the pancreatic ductal cells, they will secrete bicarbonate. So a lot of bicarb. Now these duodenal cells, these cells here, they also have the ability to secrete a lot of bicarb as well. So therefore, the small intestine is dominated mainly by bicarbonate. There's a lot of bicarb inside the small intestine. And the reason for that is because you want the pH of the small intestine to be very alkaline. So the pH of the small intestine should be around 8 to 9, whereas the pH of the stomach is about 2.5. So therefore, you have two immediate environment of your digestive tract that has high acidity and high alkalinity, right? So the chyme coming from the stomach, highly acidic. As soon as that chyme enters the small intestine, the duodenal cell sees that or senses that. It tries to neutralize the chyme or neutralize the acidity, but in order to raise the pH and maintain it at an alkaline level, it also stimulates other cells, in this particular case, the pancreatic ductal cells, to also secrete bicarb. So together, these cells and the duodenal cells secrete the bicarb to neutralize this chyme and to raise the pH inside the small intestine to about 8 to 9. Because we'll see that the enzymes of that works inside the small intestine works optimally at a very high pH. Okay? All right, so pancreatic ductal cells stimulated by this hormone called secretin that's secreted by the duodenal cells to secrete bicarb, which then helps to neutralize the chyme
and raise the pH of the small intestine to about 8 to 9. Okay? Now, secondly, you want to now stimulate the secretion of your digestive enzymes. So the digestive enzymes are secreted by another type of cells called the pancreatic acinar cells. And to help the pancreatic acinar cells secrete those hormones, there's another hormone secreted by these cells, and we call this cholecystokinin. Okay, or CCK. So cholecystokinin is another hormone. That hormone is also secreted by the duodenal cells in response to this incoming chyme that contains all that, carbon sources. Cholecystokinin will stimulate the pancreatic acinar cells. Okay. So the pancreatic acinar cells will secrete a lot of different hormones. There are 3 that I'll list for you guys. The first hormone secreted by the pancreatic acinar cell is referred to as pancreatic amylase. Remember, this is an enzyme. Did I say hormone? I meant enzyme. So this enzyme called pancreatic amylase will be secreted by your pancreatic acinar cells, which means that the pancreatic acinar cells are exocrine cells, whereas these duodenal cells that detect that chyme are your endocrine cells. Right? So pancreatic amylase, very similar to salivary amylase, but because the salivary amylase are secreted from your salivary glands, whereas the pancreatic amylase are secreted by your pancreas. They do the same thing, but in different regions of the body. So pancreatic amylase, its function is to chemically digest less complex carbohydrates into monosaccharides. Simple sugar molecules, okay, like glucose and things like that. Okay, so that's the job of pancreatic amylase. The second enzyme that is secreted by the pancreatic acinar cells is an inactive enzyme we call trypsin or trypsinogen. Okay, so it's inactive. Anytime you see this term ogen or nogen at the end, it indicates the inactive form of that, right? So pepsinogen, trypsinogen, fibrinogen, and things like that, right? So trypsinogen is activated by an enzyme that is found on the surface of the duodenal cells. So imagine the duodenal cells has these particular, let's say the surface of these cells kind of look like this, right? And they're like columnar cells. And embedded onto the surface are a bunch of enzymes. These are integral proteins, and these enzymes are called enterokinase. Well, the category of these enzymes we call the brush border enzyme. But one of the main brush border enzymes responsible for activating trypsinogen is called enterokinase. So, enterokinase. And as all kinases go, what it does is it takes a phosphate from ATP and it sticks it onto an inactive protein so that it can activate that protein. So what it does is it activates trypsinogen into an enzyme we call trypsin. And the job of trypsin is the same thing as pepsin, but trypsin works in the small intestine, whereas pepsin works in the stomach. They work in entirely different pH environment altogether. What happens to this pepsin as it enters the small intestine? Is it still functional? No, because the pH environment has drastically changed. So what would happen to all the pepsin? Yeah, it becomes denatured. Okay. What happens to the salivary amylase as it reaches the stomach? It also becomes denatured. Because what I didn't mention is that the environment inside the oral cavity has a pH of about 7. It's a neutral pH environment. Okay. All right, so trypsin, its function, very similar to pepsin over there. So function is to chemically digest less complex proteins into simple amino acids. The actuality of this is that trypsin is going to break these less complex protein into mainly like double amino acids. So amino acids, 2 amino acids that are still bound to each other, right? Typically 2 or 3 or very, very small amino acid compounds, which then this other enzyme that is embedded onto the apical surface of these cells of the small intestine that will further break down these amino acids into its simplest form. But we don't really need to get into that because they have very specific names and there's a lot of them to worry about, okay? So therefore, for us eventually, we understand that trypsin is gonna break down that less complex protein that pepsin has already broken down, and then taking these less complex protein, trypsin will eventually break them down into simple amino acids. Make sense? Okay. So that's that cell. All right, there's a third enzyme that is secreted. So this is the second enzyme secreted by the pancreatic acinar cell in response to cholecystokinin. Third enzyme secreted is an enzyme that helps to break down triglyceride. Okay? And this enzyme comes from your pancreas, so we call this pancreatic lipase. Now, pancreatic lipase can break down your triglyceride. So imagine triglyceride is a nonpolar compound, right? It's a lipid. So anytime you have a lipid, because they're nonpolar, they don't like to interact with water, they all stick together. So now if you consume a lot of lipid in your diet, as it forms this bolus and chyme eventually entering the small intestine, it basically is now delivered as a package of fat, let's just say. Okay, there's some degree of breaking that apart inside the stomach, but not as much as you think it would. So therefore, by the time it reaches the small intestine, imagine it's just a blob of fat, right? So a blob of this triglyceride. So a bunch of triglycerides stuck in here. So here's your lipid, and this lipid is made up of triglyceride. So if there's nothing happening to help break apart this lipid into smaller lipid components, then pancreatic lipase can only break down the triglycerides that's on the outside, leaving the rest of that triglyceride untouched, basically, as this lipid particle moves through the rest of the small intestine. So to help break this lipid down to smaller components in the liver, the cells of the liver produces a compound we call bile. So it secretes bile. And that bile is stored by your gallbladder. Okay, so bile is secreted by the liver, stored in your gallbladder. Okay, so therefore, when this chyme comes in, and let's say this chyme has a lot of lipid, then the duodenal cells will detect that, releases cholecystokinin, and cholecystokinin will also stimulate the liver and the gallbladder. Stimulate the liver to make more bile, stimulates the gallbladder that stores all these bile to release the bile into the small intestine. And bile is green, dark green, so therefore it's green. Okay. The bile is going to essentially attack this large lipid structure and break it down into smaller lipid structures. Make sense? So we call this emulsification of the fat. So bile emulsifies the lipids to turn this big droplet of lipid into smaller particles of lipid, which means more triglyceride are now exposed to the activities of pancreatic lipase. So the function of pancreatic lipase is to chemically digest your triglyceride into what we call a monoglyceride and 2 fatty acids. Okay. So imagine if you take this compound and you kind of visualize it under the microscope, the triglyceride you guys remember the compound is 3 carbons, right? And each of those carbons are bound to a very long fatty acid. Okay, so this compound is called glycerol, and these 3 are called fatty acids. So here's the glycerol, and here's the fatty acids. So because there's 3 fatty acids bound to a glycerol compound, we call it triglyceride. Okay. Now, pancreatic lipase, what it does is that it will hydrolyze 2 of these, and it frees up 2 of these fatty acids. So the result of this is that it will break it down into this 3 carbon compound that is still bound to 1 fatty acid. That's called a monoglyceride now, because it has 1 fatty acid attached to the glycerol compound, plus 2 separate fatty acids. Does that make sense? Okay. And then the cells of the small intestine will take this monoglyceride and fatty acid into itself through absorption and actually reconstitute it as triglyceride and package around a vesicle to deliver it, and this vesicle is now known as a chylomicron. Okay, you guys will learn all of that more in maybe nutrition class or whatever the case is later on. Okay, so up until this point, this is what you need to know. The function of pancreatic lipase is to chemically digest. Oh, I forgot the T in digest. Sorry. Digest triglycerides into a monoglyceride and 2 fatty acids. Okay.
So bile breaks down lipid into triglycerides?
No. So imagine you have a big goblet of lipid.
Yeah.
And then made up of multiple triglycerides. So sort of imagine you guys are all triglyceride, right? Now, if as you enter the body, all you guys are like hugging each other. Okay. So therefore, some of you guys are inside this, this circle. Some of you guys are outside this, whatever this thing that you guys have made. If I release out the pancreatic lipase without the bile, then the pancreatic lipase can only attack the triglyceride on the perimeter. It cannot do anything to penetrate into that big, goblet of fat. But if I also release bile from the gallbladder, then that bile will penetrate and then separate all you guys. Therefore, now freeing up the triglyceride individually so that the lipase can attack each individual triglyceride. So you can break down that into a monoglyceride and fatty acids. So to be more expedient, to be more efficient, right, in breaking down that triglyceride. Otherwise, if you don't have bile, then you can only break down the one on the perimeter, leaving the rest of it to move through the rest of your digestive system. Once that thing leaves your small intestine, you cannot break down any further amount of triglyceride. So what then happens if you don't have enough time, or if you have inadequate amount of bile, or if you have inadequate amount of pancreatic lipase to break down your triglyceride? What's going to happen to all of that remaining triglyceride? Where's it going to end up? In your feces. how do you know you have a lot of triglycerides in your feces? Lower density, your poop floats. Okay? Your fecal material is very, very highly dense, packed with like undigested material. So typically, normal poop will sink. Go home, poop tonight. After you poop, look at it. It's your poop. There's nothing wrong with it. Please don't like call people like, hey, you know, look at my poop and stuff like that. Do it on your own. All right? Don't take a picture. I don't want to see it. Nobody needs to see that. But just look at it and assess it yourself. Look at your poop like, it sank. All right, cool. What color is it? You know, is it nice and like kind of tannish, brownish or whatever? Or does it have streaks of blood on it? Does it have streaks of black? Is it very black or is it, you know, normal color? Is it green? Because it can be if you have too much bile secretion, you eat too much of, you know, certain types of vegetations and stuff like that, right? It may cover your fecal material and kind of turn it a different color. But if it's a black poop, you need to worry about it because that can indicate internal bleeding. Okay, we talked about that already, right? If it floats and it smells really bad, that's because of the fat that's inside your fecal material. Okay, so therefore, if it floats, that means you now have malabsorption of your triglyceride, which means you don't have enough triglyceride being taken into your body. You don't have enough of that stored inside your body, which potentially is very bad because triglyceride is very important for a lot of different things that we use throughout the whole body. Okay? Make sense? So what happens if you don't chew your food adequately at the beginning to break down that nice little steak that you have, to break down all the carbohydrates, to also break down that lipid that you basically consume, right? Then whatever the salary amylase is that helps to break down the carbohydrate, it's not going to be able to break down adequately the complex stuff into the less complex stuff. So a lot of that will remain more complex overall. As this bolus enters the esophagus, the salivary amylase continues to do its work because the esophagus cell has a pH of 7, so therefore nothing's happening to that enzyme. By the time the bolus reaches the stomach, salivary amylase becomes denatured because of the high acidity of the stomach. Now, in here, you start to secrete all of these stuff. Pepsinogens convert into pepsin, so therefore pepsin starts to chemically break down your protein structure. But what if you don't have adequate amount of acidity inside the stomach? What if, like Lindsay says, you start to pop antacids like it's, you know, Halloween candy or whatever it is, right? You decide you like that chalky, fruity taste that's offered by your Tums. You're like, this tastes really good. This is gonna be my main diet for the rest of the week or whatever the case is, right? And you start popping pills like, you know, your life depends on it. Well, then you will reduce the acidity in your stomach drastically. It's still acidic enough to break down or to denature your salivary amylase, but it's not acidic enough to activate an adequate amount of pepsinogen. So the amount of pepsin that you have decreases. If you have less pepsin, you will have less chemical digestion of your complex proteins. So therefore, by the time the chyme reaches the small intestine, all this stuff is happening. But even if you secrete all of these enzymes into the small intestine, you still have a lot of complex carbohydrates that the pancreatic amylase cannot digest adequately into monosaccharides. So you will lose a lot of your sugar in your feces, okay? If you cannot break down your complex protein adequately in your stomach, by the time trypsin is activated, trypsin will try to, but will fail to some degree to break down all of that complex proteins into amino acids. So you will waste most of your protein in your fecal material. And obviously, if you don't do any of that stuff, and let's say you have inadequate amount of bile secretion, you're not gonna have enough emulsification of that fat, and therefore your pancreatic lipase will not be able to chemically break down the triglycerides adequately. So all of that leads to what we call malabsorption, because this is the inability of your digestive system to adequately digest all this food to the point where the cells of the small intestine can absorb all of the simplest compounds into your body. So what's one of the things that may happen if you have a bunch of carbohydrates left undigested that goes into the fecal material?
what's gonna happen with that? What's a condition, what's a reaction of the body if you have a bunch of, let's say, polysaccharide or disaccharides or whatever, like some sort of complex carbohydrate still lingering within your digestive tract. What do you think? It's not in blood, so you don't have to worry about diabetes. You just lose it, right? The sugar has very high osmotic effect. So therefore, the sugar is now floating around, and a lot of them are in the lumen of your large intestine. It's gonna start to draw a lot of water into the large intestine. And therefore, you end up having diarrhea or some kind of loose bowel movement, okay? Of course, you lose all of that, carbohydrate, which is the worst thing. The diarrhea is just whatever, it's just a reaction of that osmotic effect. It's the protein also that's very important. You lose all of that complex proteins, you will not have adequate amount of amino acids to make your protein structures inside your cells, okay? So therefore, you become protein deficient. You lose triglyceride, you lose the fatty acids, you lose the ability of your cells to massively make ATP when you're at rest. So if the cells cannot use fatty acids to make ATP, it starts to use glucose to make ATP. And if you deplete your glucose supply when you need it, when you're exerting yourself, you won't have enough glycogen supply or glucose supply, and therefore you will be chronically fatigued as well. So everything adds to some kind of outcome later on, okay? Now, what do you guys think is the #1 cause of malabsorption in the United States? It's not the ability of us to chew food adequately, right? We have plenty of food, right? I mean, if you ever need food, if you don't want to cook or whatever, I mean, I just found out they have instant rice. What the fuck? That's crazy, right? They sell it at Costco for a box that has 20 pack. All you have to do is take it home, take it out, put it in the microwave, press a couple buttons, and boom, within a couple minutes, instant rice. You don't even have to buy a damn rice cooker anymore. You can have rice on the go, just like that. It's wonderful, but it makes us lazy. So therefore, there's no need for us to basically be deficient in any kind of, consumption of these food. But we do have some degree of malabsorption. What do you think is the main cause of that? It's leading up to be one of the most common causes in the United States now.
Probably the small intestine.
Which part of the small intestine? And it's not really actually the small intestine itself, it's an accessory organ that releases certain contents into the small intestine.
Pancreas.
The pancreas. So this is due to what we call pancreatic insufficiency, which means for some reason the pancreas is not stimulated enough to release those enzymes. And even if those enzymes are released into the pancreas, for some reason they are not adequately activated. And because of that, you have now the inability or a drastic decrease in the ability to chemically break down your food compounds. So therefore, a lot of them stays in this complex form, and it will eventually be excreted out of the body. Right. So, don't know why that is. It's just one of the phenomena. It's a shift that's going on that we see a lot more frequently. Right now, of course, you can actually also cause some kind of alteration in the mucosal surface of your small intestine, especially if you have things like celiac diseases and other stuff where you can't digest the grain and all that. It causes some degree of inflammation and all that good stuff. But whatever, that's no longer the most common cause. Okay. So anyhow, that's happening inside the small intestine. Okay. Keep in mind, for every enzyme that we talked about, know the source that produces the enzyme. What produces the enzyme? So for example, salivary amylase. What produces salivary amylase? Well, your salivary glands. Which one? Doesn't really matter, right? You pick your favorite, except for sublingual. Don't pick that one. That one does not produce salivary amylase. So either submandibular or parotid gland. Most likely parotid gland if you want to get more technically correct. But it doesn't matter, right? Just say salivary glands altogether. That's fine. Okay, later on you have to be a lot more specific, but for now, for us, it's okay. You don't have to be that specific. So, what produces salivary amylase? Your answer should be salivary glands. Okay?
where does salivary amylase work? So what is the site of action of these enzymes? They never ever work in the glands that secrete them. So the salivary amylase should not be functional inside the salivary glands. That's weird. Now it's going to destroy the structure of the salivary glands. So you don't want that to happen there. You want the salivary amylase to be active once they are secreted into the oral cavity. So the site of action of salivary amylase is within the oral cavity mainly. Now you can say, well, as the bolus comes down the esophagus, salivary amylase is still working there, so fine, you can add that also, okay? But the major site of action of salivary amylase is in the oral cavity. And then what's the function of salivary amylase? Well, the function is listed. It is there to chemically digest complex carbohydrates into less complex carbohydrates. Okay? So know those information if I ask you to identify what is the source that produces those those enzymes. Where do those enzymes work? Sort of like the source of it is your home, and where you work is your work, right? Unless you work at home, then this example is basically no good. Okay, so maybe not that. Anyhow, so where do these enzymes work? What site inside these digestive tract does it work? And especially true for, let's say, things like in the small intestine. So pancreatic lipase, what is the source that produces pancreatic lipase? And you have to be very specific on this one. Because you can't just say the pancreas. Because the pancreas has a lot of different parts, and one of the parts of the pancreas is an endocrinic portion of the pancreas, which we didn't really talk about. So therefore, the specific area in the pancreas that produces these pancreatic lipase is known as the pancreatic acinar cells. Okay. So pancreatic acinar cell produces the pancreatic lipase. Where do the enzymes work? Where do they work?
In small intestine.
In the small intestine. So that's the site of action of pancreatic lipase. And what's the function of it? Its function is to chemically digest triglycerides into a monoglyceride and two fatty acids. What is the stimulus of the pancreatic acinar cell to secrete pancreatic lipase? Yeah, cholecystokinin. What is the source that produces cholecystokinin? The duodenal cells. What is the stimulus of the duodenal cells to secrete CCK or cholecystokinin? The acidity of the chyme. Okay. Start to work yourself, connect all these dots together. That's why all these arrows are there for you guys to connect it together. Once we get to the endocrine lecture, there's going to be a huge amount of arrows connecting everything together. Right? I call that the GPS system of the endocrine system. Okay, so therefore, make that list that I asked you to make for the endocrine system, but also follow along with this GPS mapping of where the sources of these hormones are and where they go to and what they do at those particular targets and stuff like that, and what is the stimulus of the sources to secrete those hormones and so on and so forth. So same thing for enzymes too. Think about the source that produces these enzymes, right? And also think about where these enzymes work, and also think about the stimulus that stimulate those sources to produce and secrete those enzymes. So my question to you is then, what is the source that produces and secretes pepsin?
The chief cell.
There'll be none. Because the chief cell secretes pepsinogen. They don't secrete pepsin. So therefore, pepsinogen is converted into pepsin. So, okay. So technically, you can say the chief cell. It's okay. Because then it's like sort of like, well, you know, pepsinogen is inactive anyway, so you have to activate it. Well, yeah, but if I say, okay, well now the pH of inside your stomach is no longer 2.5, you're not going to adequately activate enough pepsin. Okay, so there's a lot of condition that kind of follows with all of that. Okay, good. So that's that. All right. So now, whatever undigested food that you have within the small intestine, and the small intestine has 3 portions of it, right? And it's long. It's long so that you can have enough time to chemically break down all this food, so all the cells can absorb this food adequately. But if for some reason, you don't have adequate amount of enzymes that is active, you don't have all of these things that basically is secreted out, then you have a lot of undigested material that goes into the large intestine. So large intestine absorb very little. It mainly regulates the amount of water in the intestine. Oops. How can I spell? Okay. So large intestine, okay? Large intestine is also lined with simple columnar epithelial tissue. So because it's continuous with the small intestine, it's the same thing. The large intestine is not highly folded and stuff like that, so therefore it doesn't do a lot of absorption. One of the main things that it does is that it will help regulate the amount of water within the lumen of the large intestine. Okay, so first and foremost, regulate water within large intestine. If the undigested material is maybe too aqueous, let's just say, then the cells of the large intestine can absorb some of that water away from that fecal material, from that undigested material, right? So that you don't have loose bowel movement. If the fecal material or undigested materials within the large intestine is too dry, then the cells of the large intestine can secrete some amount of water to mix with that undigested material so that your bowel movement is not inhibited, which then leads to what we call constipation. Okay? So by regulating that amount of water, it is allowing for adequate amount of excretion of that fecal material, okay? Without you straining too much and stuff like that. And you shouldn't be straining that much anyways. Anyhow, so now also living within the large intestine, so this is one of the main functions. It has other functions too, but this is the only one that I really care for you guys to know about. Living within the large intestines are a bunch of bacteria, okay? The bacteria that lives within the large intestine is referred to as your microbiota. And one of the great things about the microbiota is that it can digest some of these undigested food materials that we can't digest. So we are providing some of the nutrients to these microorganisms. Okay, so the microbiota is just essentially a set of microorganisms that lives within your large intestine. Now there's different types of bacteria in there. So another way for us to kind of control the population of this bacteria is to allow them to compete for the same resources that we provide. So they basically fight off each other to kind of regulate their own population, okay? Our immune system can also help to regulate some of the population of these microbiota as well. We don't want too much overgrowth, we don't want too little of them, because if that's the case, you might not have enough of the benefits that comes from these guys. Now, oftentimes, the microbiota is gonna live harmoniously or symbiotically with the cells of the large intestine. So in this symbiotic relationship, there's 3 different symbiotic relationships that can be achieved. The first symbiotic relationship is what we call a mutualistic relationship. So the microbiota can achieve a mutualistic relationship with the large intestine. In a mutualistic relationship, just imagine, right? You guys starting a new relationship, everything is nice and good. It's a honeymoon period. You know, you buy flowers for each other, you buy gifts for each other, you say, I love you every single time you get off the phone or whatever the case is, right? You say, oh yeah, I miss you, think about you all the time, blah, 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 all that, you know, soppy BS that we all talk about, right? So that's a mutualistic relationship. Both parties benefit, okay? So these microbiota benefits from all of the undigested material, but they also make certain things that benefits us. And one of the compounds that are made in this mutualistic relationship that benefits us is vitamin K. Okay, and we know vitamin K is very important for the clotting purpose of our blood. Okay, so that's a mutualistic relationship.
okay? Now, the second type of symbiotic relationship that is achieved by certain microbiota and our large intestine is what we call commensalistic. In a commensalistic relationship, imagine you've been together with this person for like 5 years, right? Eventually, without any fail, one person will do slightly more than the other one, okay? So let's say, you go through this relationship or these microbiota, it's the bacteria are still receiving all the benefits from the undigested materials that we provide for them, but they no longer make anything that benefits us. But they don't do anything to harm us. So essentially imagine your relationship now is like, 3, 4 years, 5 years down the road, you're still living together, you're still happy, but one person now cooks and the other person's supposed to clean, but they don't. They're like, I'm tired. So the other person says, all right, fine, I'll help you out. So they start to clean too. Eventually that becomes a habit. So therefore only one person does everything and the other person just kind of reap the benefits without adding any benefit to that relationship. So it becomes more or less a one-way relationship, right? But both are still happy. There's no destructive forces that's working behind all of these nefarious activities yet, okay? But eventually, all relationship comes to an end. So therefore, there's a third type of symbiotic relationship achieved by these microbiota with the large intestine, and we call this a parasitic relationship. Okay, in a parasitic relationship, for some reason, let's say your immune system is not strong enough to basically correct or maintain the population of these microbiota, or one of these colonies of microbiota has been wiped out and therefore there's only one remaining one, so they start to flourish out of control. If you allow these bacteria to flourish out of control, they will start to cause harm to your intestine. Okay, so therefore, in this parasitic relationship, if you guys are taking microbiology, any kind of microbiota or any kind of bacteria or foreign entity that lives within us symbiotically can become what we call a pathogen. Okay, and we call that an opportunistic pathogen because they now have the opportunity to start to grow out of control, consume more nutrients than actually is allowed for them to consume, and start to take over normal functions of that part of the body. Hence, they become parasitic. Okay? So just keep in mind these three. That's the lesson in life. Just kidding. Don't be morbid. Like me. Once you get to my age, you can be jaded. You guys are still young, so live life. Think about love as love, right? Don't think of it as a chemical imbalance. Okay. So now, once you have all this undigested material going through your large intestine, eventually it gets deposited into your rectum. So imagine your rectum. Okay, so here's the rectum, here's the lumen of the rectum. Okay, it has all of these little Holstra, structures. So here's the rectum. And the rectum is still lined by simple columnar epithelium. But the rectum eventually narrows into a canal. And we call this the anal canal. And the anal canal is going to end as an opening we call the anus. So here's your anus. Okay? So now imagine fecal material entering your rectum. So here's feces entering your rectum. As the fecal material is deposited into the rectum, it causes a stretch. So we'll put this in green because it's a stimulus. So it causes a stretch of the rectum. When the rectum stretches, a sensory nerve detects this and will relay this information to your spinal cord. So here is the lumbar spinal cord. Here is your sacrospinal cord. Okay? So the stretch is being relayed back to both the lumbar and the sacrospinal cord. So these are sensory nerves. Okay. Now you think about it, right? So this fecal material here, let's say now it's kind of moving here, so you have feces right here, causing that stretch along the way. Oh, another thing I forgot to mention is that there's a bunch of smooth muscle surrounding these rectal area, the sigmoid colon and rectal area, and they will converge right along the pathway of the anal canal. So all of these particular, so let's say imagine you have this muscle all converging right here, and when muscle converges, it forms what we call a sphincter. Okay? So very similar to your urethral sphincter that we talked about. This sphincter is known as your internal anal sphincter. And the internal anal sphincter is smooth muscle because it's a continuation of the muscle that surrounds your rectum. So smooth muscle. Okay. Now, the anal canal, as it becomes the anus, has to pierce through the pelvic diaphragm floor so that it can exit to the outside. So therefore, it pierces through this muscular floor here. And this muscular floor is made up of skeletal muscle that we call the external anal sphincter. Which is skeletal muscle. Okay. All right. So as these stretch receptors along the way detect the stretch of the fecal material being moved throughout this rectum toward basically the entrance of the anal canal, the sensory nerve sends these signals back to both the lumbar spinal cord and sacrospinal cord level. From the sacrospinal cord level, it's going to stimulate a nerve. And this nerve goes all the way back to, let's say, the smooth muscle of the rectum to stimulate the smooth muscle of the rectum. Okay, this is your parasympathetic nerve. Okay, so contraction of smooth muscle. Okay, so fecal material enters the rectum gets stretched. What do you want to do? You want to start contracting, right? The fecal material. Now the fecal material is now going to enter the anal canal, but in order for it to enter the anal canal, you have to relax this internal anal sphincter. So therefore, this sensory nerve that is now being relayed to the lumbar spinal cord level is going to stimulate another type of autonomic motor neuron, and this is going to induce relaxation of the internal anal sphincter. And this is your sympathetic nerve. Okay? So in this particular reflex, the parasympathetic nerve stimulates the contraction of the smooth muscle in the rectum, pushing the feces into the anal canal. But in order for the feces to enter the anal canal, the sympathetic nerve that controls that sphincter has to induce relaxation. So now you have relaxation of your internal anal sphincter. The feces enter your anal canal. Okay? But every single time you have this sensation, this stretching sensation, the sensory nerve that relays this information back also relays this information all the way up to your cortex, your brain. So that you can be aware of that particular sensation of stretch in your rectal area, right? So the cortex says, okay, cool, thanks for letting me know. I'm gonna relay this information all the way back down to the sacrospinal cord level. And from there, I'm going to stimulate a somatic motor nerve. And this somatic motor nerve will stimulate the external anal sphincter, so somatic nerve here. Okay? So now you get to choose what you want to do. Are you in an area that it's okay for you to kind of poop, or are you not? And even if you go to the bathroom and you sit down and you want to excrete out or get rid of this fecal material, you still need to consciously relax your external anal sphincter. Okay? So you can induce relaxation to allow for pooping to occur, or if you're in class or you're in a car and you don't want to poop in front of everybody, then you need to contract your external anal sphincter. If you contract your external anal sphincter, pressure emanates and pushes the feces back into the rectum. And this whole reflex is inhibited until more feces enter the rectum. And as more feces enter the rectum, the stretching becomes a lot more. And therefore, there's a higher urgency that you need to get rid of this feces, right? Then eventually you're gonna start to prairie dog, and then you have to go to the bathroom and then, get rid of that stuff. We call this the defecation reflex. Okay. And now we come to the end of our tour. Please wash yourselves as you exit this fecal-filled tiny little bus as we rinse down the bus for the next tour to come. That's disgusting, right? Somebody had to swallow that bus and then, don't want to think about it. Any questions about the digestive system? No? All right, that's it. Let's go to lab.